This lesson is for Module 11 in biology, and it's going to be the first part of that module. So this module is dealing with the invertebrates of the animal kingdom. So before we get into the invertebrates, let's briefly talk about how kingdom Animalia. So we are now on the fifth kingdom. So we did kingdom Onera, we did kingdom Protista, we did kingdom Plantae. Then we did kingdom Fungi. Then we took a break and did some studies of the cell and chemistry and reproduction. We did a unit on ecology and a unit on evolution, and now we're back to the last kingdom, which is kingdom Animalia. We divide kingdom Animalia into two main categories. The first is the invertebrates, and then secondly are the vertebrates. The invertebrates are those animals that have no backbone. And the vertebrates have a backbone. So every kingdom in kingdom Animalia except for phylum Chordata makes up the invertebrates. So and actually that's not totally true, because there are a few members of phylum Chordata that are invertebrates, but basically all the vertebrates fall into phylum Chordata, and that's the only phylum among the vertebrates, whereas the invertebrates are all the other phyla, all other animal kingdom phyla. And as I said, there actually are a few invertebrates that are members of phylum Chordata, but the vertebrates are all members of phylum Chordata. Okay, so invertebrates and vertebrates, no backbone, backbone. That's how we divide the animal kingdom. Another thing that we talk about when we talk about the animal kingdom is something called symmetry. And symmetry is looking at how you can divide an organism into two identical halves. So let's look at some of the types of symmetry that we have. So this figure shows symmetry in people, and this is showing that in people we can draw a line down the center of that person, and we end up with identical right and left halves. So that type of symmetry is called bilateral symmetry. And there are other ways that you could divide a human. So we could draw a line and divide between front and back halves, but those halves would not be identical, obviously. You could divide a line here and get two halves, but again, they would not be identical halves. The top is going to be different than the bottom. So bilateral symmetry, where you get equal right and left halves. So humans have bilateral symmetry. However, so besides bilateral, so that's bilateral symmetry, is right and left halves. Other types of symmetry are called spherical and radial symmetry. So with spherical symmetry, if an object if an object has spherical symmetry, you are able to draw a line anywhere, any place through that. If you make any cut that goes through the center, you will end up with two identical halves. If this were a perfect circle, I could draw a line like that, and that is spherical symmetry. If I drew a line like this, so any line that goes through the middle, so I could erase this one now, any line that goes through the middle is going to lead to two equal halves. If I drew one like this, that's spherical symmetry. Any line that goes through the middle results in two equal halves. So let's look at a figure that will show that. So on the left here is spherical symmetry, and you can see, as I've drawn before, any line that goes through the center here, any line that crosses through the center will lead to spherical symmetry. Okay, then another type of symmetry is radial symmetry. And in radial symmetry, you can make any longitudinal cut through the center, and you will end up with two identical halves. So it's a little bit hard to see, but come on. If I take and I cut this sea anemone down through in that way, then I will end up with two halves that are identical. Or so it has to be a longitudinal cut, or if I cut this way, so if you can think of cutting through a plane in that way, again, you would end up with two identical cuts, or two identical halves. So this has to be a longitudinal cut. Through the center, and then you will end up with two identical halves. So spherical, it can be any cut through the center. With radial symmetry, it needs to be a longitudinal cut through the center. And then we've already seen this bilateral symmetry, so this is a flatworm. Any cut down through the middle that results in identical right and left halves is considered bilateral symmetry. So one of the things that we look for in looking at members of the animal kingdom is what kind of symmetry do they have. So there are some on your own questions on your own. 11.1 lists some organisms, and we can look at those. The first one is a manta ray, and a manta ray would have bilateral symmetry. You could make a cut and end up with identical right and left halves. The next one is a sponge, and the sponge would have no symmetry. So if you get an organism that is irregularly shaped, then there's no way that you can cut it to get two identical halves, and a sponge is one type of that. Another example is the jellyfish. And a jellyfish would have radial symmetry. So you can make any cut through the middle, any longitudinal cut through the middle of a jellyfish, and you would end up with two identical halves. And then lastly is a coccolithophore. So you see, that's two C's. Coccolithophore. And it's a spherical or shaped organism, and therefore it would have spherical symmetry. You could draw any cut through the middle of this organism, and you would end up, I didn't draw that very well, but you would end up with identical halves for any cut that goes through the middle of the organism. Okay, so be sure you are familiar with those different types of organisms within, or different types of symmetry within the animal kingdom. Okay, so in the animal kingdom, there are about 20 different phyla, but we are going to look at, I believe, only 5 in this chapter, and I think we will get to 5. Yeah, so we're going to deal with 5 phyla. As you're thinking about these different phyla, the main thing I want you to be able to do is to, if I give you the name of an organism that we have talked about, I want you to be able to place that organism into the correct phyla. Okay, so the first phyla we are going to deal with is called phylum Porophora. Or Porophora. Phylum Porophora, and the organism that make up that are contained within this phylum are sponges. Most sponges live in salt water. There are a few freshwater sponges, but the majority of them live in salt water. So therefore, we would say they are, the term that's used for organisms that live in salt water is that they are marine mammals or marine animals. Members of phylum Porophora, as we just saw an example of this, they have no symmetry. They can vary in size from being very small to very, very large, but they tend to be irregularly shaped, and so they have no symmetry. They are made up of 2 layers of cells. And those 2 layers are called the epidermis So the epidermis is an outer layer of cells, Your skin is called your epidermis. The outer layer of your skin is called the epidermis. And so it's a similar type of structure. It's the outer layer, and its main job is for protection. So it protects the inside of the organism. And then there is another layer of cells that is called the mesenchyme. And the mesenchyme, it's kind of a jelly-like substance that is between the two layers. So this is kind of a jelly between two layers. And then you have the inner cells inside of that make up the sponge. So outer layer is the epidermis. Then you have this layer in between that separates the epidermis from the inner cells, and the inner layer is called the inner cells. If we look at this layer that is called the mesenchyme, so let me write that up. So this in-between layer that is called the mesenchyme, there are two types of cells that are produced within those, and so within the mesenchyme, there you will find spicules and you will find spongin. The spicules are primarily made of calcium carbonate, which you might have heard of. It's also referred to as lime. So it is a it's harder, it's more prickly. So this is kind of harder, prickly. So the spicules really help to give the sponge its support. Then there is also a kind of a web of protein that is called spongin, and this tends to be more soft, not so hard and not so prickly. And so most of the sponges that we use today for cleaning are artificial sponges, but we used to use natural sponges for cleaning and for bathing, and usually the ones that we would use for bathing or cleaning they contain this spongin because it is it's a softer material. Okay, so spongin and spicules are present within sponges and give it either the hard feeling or the softer feeling of a sponge. The cells that make spongin and spicules are called amoebocytes. and these amoebocytes do other things for the sponge as well. But so the main functional cells of a sponge are called the amoebocytes. Let me see, let me see if I can find a figure that is not in your book, but I'll see if I can find it. Maybe I won't be able to. Let's see, let's look here. Yeah, so this is the figure I wanted you to see. So this is a drawing of some of the parts of a sponge, and some of the parts that your book points out are the collar cells. So you can see the collar cell right here, up here on the top, and the interesting thing about the collar cells is that they have flagellum. So let me go ahead and write that up on the board. So there are collar cells, and these collar cells have flagellum. So remember, those are those tail-like substances that come out. And what these flagellum do is the flagella are constantly beating the water, and as they do that, they are pulling the water into the sponge, and then the sponge is able to remove algae or bacteria from it that they use as a food source. So the flagella on those collar cells are really important for the sponge's food source. Let me see, another figure, let's go back to look at the same figure here. Another thing I want to point out to you, so sponges have pores, so remember they're called porifera. So the term porifera comes from the fact that they have these pores, and that is the way that water gets into the sponge. So water, you can see these arrows here pointing that water flows into the sponge through the pores. Well, you can imagine that if a sponge keeps having water go into it but not going out, that sponge would eventually swell up to the point and it could actually explode. So there is actually another opening in the sponge. So we have pores that allow where the water enters. So there's pore cells, and water enters in through those. We already saw that the collar cells also pull water toward the cell. But then we have some cells that are called the osculum, and it is through the osculum that water leaves a sponge. So that if the sponge is taking on water, it also needs to have a way to get rid of the water, and that takes place through the osculum. Remember we talked about those amoebocytes, and I said that they are the main functional cell of a sponge. So once food is brought into the sponge, then the amoebocytes have pseudopods. And remember, those are those false feet. So with those pseudopods, they are able to digest the food, and it also then gets transported so that they digest the food, and the food gets transported to where it is needed. And then the amoebocyte, so they transport the food to where it is needed. They also transport the waste. So once the sponge cells have used the food, they will produce waste, and they transport the waste to the epidermis, and then through the epidermis, the waste gets eliminated. So these amoebocytes are pretty important cells. They have a lot of different jobs that they do. Besides transporting food, they will also transport oxygen to the cells. So they'll transport oxygen. And as we already said, they also they make the spicules and spongin. So the amoebocytes, the functional cell of a sponge, they do a lot of things that are important for the functioning of the sponge. Okay, the next thing we need to talk about with regard to the sponge is how do they reproduce? And we will see that there are several ways that reproduction can take place. They can reproduce asexually or sexually. So let's first look at how they can reproduce asexually. So asexually, one of the ways that they can reproduce is by regeneration. And we'll begin to see this now in other organisms. So regeneration means that if you cut away, if a portion of it gets cut away, that portion will grow into a new sponge, and then the sponge from which the piece was cut away will also regrow that part. So from one sponge you get two sponges. And this is an important way for sponges to be able to reproduce. There was a time when so sponges will eat clams. Interestingly enough, they are able to they'll eat clams, and there were some like marine farmers that were growing clams and the clam or harvesting clams, and the sponges kept eating. Actually, I'm sorry, I'm confusing that with jellyfish. So that story that I was going to tell is actually true of jellyfish. It's not true of sponges. So, but sponges do reproduce by regeneration. Another way they can reproduce asexually is by something called a gemmule. So in a gemmule, what happens is that this usually happens in cold weather. And in cold weather or other harsh conditions, what they will form is a cluster of cells in a hard shell. So this hard shell will protect these cells during the extreme weather conditions, and then when it becomes warm again, then this gemmule will break apart, and these cells can then grow into a new sponge. So that's another way of asexual reproduction, and this one is particularly done when there are harsh weather conditions like cold weather. Sponges can also reproduce sexually. So they can reproduce with sperm and egg. So the sperm and egg are both made by collar cells. And the way that sponges reproduce is that the sperm get released into the water. The eggs get released into the water. And then if a sperm cell meets an egg cell, then fertilization will take place. So, you have to assume that the sperm and eggs are going to be released, in the relatively the same vicinity and at the same time. And then it's just a matter of probability whether the sperm and egg will meet up with each other and fertilization and sexual reproduction will take place. So sponges they do have a means of reproducing sexually as well as asexually. Okay, so that's what we're going to say about the phylum Porophora, which makes up the sponges. Okay, next we are going to do phylum Cnidaria. So the C is silent here. Phylum Cnidaria. And the organisms that we would be familiar with that are part of this phyla are jellyfish, sea anemones, spelled that wrong. Sea anemones, and then there's a organism that is called a hydra. that is a microscopic organism. The thing about phylum Cnidaria is that they come in two forms. And those two forms are either a polyp or a medusa. So a polyp looks more like a disc. And a medusa kind of looks like a floating umbrella. Both of these would have tentacles, but the tentacles are more apparent in the medusa form. So when we think of a jellyfish, we usually think of, the umbrella type substance, something with the legs hanging down. So that would be the medusa form. The tentacles are more apparent. Polyp, there are some organisms in phylum Cnidaria that spend their whole life as a polyp. And then there are others that will spend part of their life as a polyp and part as a medusa. Some of the coral, so coral is another member of phylum Cnidaria. So some forms of coral spend their whole lives as the polyp form. So let's look at some of let's look some at, do I have some? I guess I don't have any. In your book on page 336, there are some examples of phylum Cnidaria. So you can see a hydra there, which is in its polyp form. You can see a sea anemone in the polyp form, and you can see coral in the polyp form. And then you can see the jellyfish, which exist in the medusa form. Just like the phylum Porophora, there are layers of cells within phylum Cnidaria. And so you have the outer layer, which is the epithelium. And then in between those two layers, you have the mesoglia. So that's like that jelly-like substance that is between the layers. And then we have the inner layer of the epithelium. Okay, so that's basically the same as we saw in phylum Porophora. If we look at within the epithelium, there are two primary types of cells, and those cells perform different jobs. So one type of cell is a nerve cell, and the nerve cell is what is able to sense outside sensations or stimuli and respond to those stimuli. But then there is another type of cell that is called a contractile cell. And the contractile cells are cells that are involved in movement. All of the, if we think about the type of symmetry that members of phylum Cnidaria would have, they would all have radial symmetry. So if you made a longitudinal cut through an organism, then you could divide it into 2 equal halves. Okay, a couple other structures that you should be familiar with is on their tentacles, members of phylum Cnidaria have these little structures that are called nematocysts. And what the nematocysts do is that so nematocysts are on the tentacles, and the nematocysts release a toxin. And that toxin can then it's injected into either their prey or their predator, so they can use it to either capture food or to defend themselves against a predator. So if they inject it into their prey, it will paralyze that prey so that then they are able to consume it, or it can be injected into a predator that is trying to capture them for food, and can incapacitate that predator so that it can paralyze the predator so that they cannot so that the member of phylum Cnidaria cannot be used as a food source. One of the interesting things about phylum Cnidaria is that they don't really have a respiratory system or a like waste system. So they do not have this, they don't have this. And the reason for that is that their cells are really, really thin. And so oxygen and carbon dioxide, so when they need oxygen, the cells are so thin that it can just move across the membrane of the cell. And when they need to get rid of carbon dioxide, which is a waste, again, the cells are thin enough that it's just able to diffuse across the cell membrane. And the same thing is also true of getting rid of waste. The cells are thin enough that the waste can just go through the cells and be eliminated from the body in that way. So they don't have a digestive tract that ends in a way that in an ending where the waste gets eliminated from the body. In terms of reproduction, the members of phylum Cnidaria can reproduce asexually or sexually. And one of the ways that they can reproduce asexually is by something that is called budding. And what happens in budding is that you get a small pouch that forms on an organism, and that little pouch grows and then eventually is detached from the parent organism, and it will grow into a new organism. So with the case of a jellyfish, you'll get this little pouch forming, and then it will then grow into it'll bud, and then it'll grow to the point that it will break off from the parent organization or the parent organism. So here is a member of phylum Cnidaria that is called a hydra. And what I want you to see on this hydra down here is that, so here's our hydra, and then this a bud can form, and then that bud will grow and it'll eventually break itself off and then become its own hydra. But they can also reproduce sexually, because within the same hydra, here's a testes that can produce sperm, and the ovary that can produce eggs, and fertilization can then take place through that means. So most of these hydra are either male or female, but there are some that are both. And so I don't know if have we used the term hermaphrodite? So when we're talking about hydra, which is a member of phylum Cnidaria, most are male or female. But some can produce both eggs and sperm. And the term that we use for those that are able to do that, they're called hermaphrodites. They can produce both eggs and sperm. And in terms of reproduction, again, the eggs and sperm get released into the water, and then it's a matter of time and place, the probability that the eggs and sperm will meet up and fertilization will take place. The other thing that I wanted you to see on the hydra, then, here's these nematocysts. So see these kind of pointed, almost thorn-like. So these are the tentacles here, and then these prickly points here are those nematocysts that would release that toxin that can then, paralyze or stun any prey or predator. Let's look at reproduction in a jellyfish. So a jellyfish is another member of phylum Cnidaria, and usually when we think of a jellyfish, we think of it as this medusa form. Okay, so what we usually refer to as a jellyfish is the medusa form. And what is true about jellyfish is that they will release either eggs or sperm. So here you can see this is a female that's releasing eggs, and then the male will release sperm. The male releases those sperm into the water, and then those sperm go to the female and fertilize the egg. Then the fertilized egg is held on the tentacles of the female until it grows, and then it will eventually be released as a form that is called a planula. And that planula will then form a polyp. And then that polyp will grow and develop. And you can see that as that polyp grows, then it begins to form these medusas. Okay, they're more disc-shaped structures. And then eventually these medusas will break off, and then you have, and the tentacles will grow, and you have what we call a jellyfish, which is really just the medusa form. So a jellyfish goes through both the polyp form and it goes through the medusa form. And again, you can't see them here, but the tentacles of a jellyfish also have nematocysts that will poison. And so when you get stung with a jellyfish, if you've ever gotten stung with a jellyfish while swimming, it's these nematocysts and that toxin that gets released. For you, it's not enough to kill you, but it will. It can cause irritation. It can cause stinging. So let me review again what happens in terms of reproduction for a jellyfish. So they are either male or female. So the males release their sperm into the water, and then those sperm swim to the female and fertilize the egg. Then the egg is held on the fertilized egg is held on the female's tentacle until it grows to the point where it is released as a planula. That planula then will swim away, and it attaches itself to the bottom, and it will grow, and now it's called a polyp. That polyp will keep growing and eventually will start to form these medusas. Then the medusa will break away. You have a new medusa, and tentacles will form, and you have another adult jellyfish. So again, the part of the jellyfish that we think of is really just the medusa form. Another thing I should mention about this polyp form is that the polyp form, so the medusa form can only reproduce sexually, but the polyp form can reproduce by budding. So when it's in this polyp form, it can form a bud, and that bud can grow and then break off. So the polyp form of a jellyfish can also reproduce by budding, or it can go through this process that we have seen before. Okay, I believe that we will stop there today because we're getting ready to go into a new phylum, and I think that's enough information for today, and we wouldn't really have time to get through the next phylum. So let's just stop with phylum Cnidaria. So we did this today. We did phylum Porifera, which are the sponges, and then we did phylum Cnidaria, which constitutes the jellyfish, hydra, the organisms that have tentacles on them.